Hello, and cell cycle mitosis and meiosis. I split into knowledge third and require practical third, and then meiosis. So let's have a look through. So yeah, we're just choosing interphase, prophase, metaphase, anaphase, telophase for these stages. So let's have a go. Yeah, so the S phase, well, that's part of interphase. Interphase is G1S and G2. When chromatids separate, well, that's during anaphase. So the centromeres are pulled apart and therefore the chromatids separate. Chromosomes relax. I prefer the word decondense than relax, because relax is more of a muscle term, but yeah, that's telophase. The stage when crossing over occurs between homologous chromosomes in meiosis, a hard one, and you don't technically need to know this, but it's prophase, and it's prophase 1, just before they go to the equator and arrange either side for independent segregation in metaphase 1. Semi-conservative replication takes place, well, that's when DNA is replicated, so that's the S phase of interphase. So I'm putting an I on for interphase there. Yeah, that's a Roman numeral 1, it's not interphase, it's a Roman numeral, prophase 1, before you tell me off. What stage is followed by cytokinesis? Well, that's telophase. Cytokinesis, you know, it's normally in process during telophase, but you would say it follows telophase really. Stage that's reduced in length during tumor formation, well, that's interphase. Yeah, you lose a G1 or a G2, as in it skips it. It's like advanced to go in Monopoly. It just, yeah, it doesn't do that stage. It goes straight to the next stage. And the enzyme that separates the two strands, so a bit of DNA replication, so that's helicase. You can put DNA in front of it. The bond broken, yeah, that's hydrogen bonds. And yeah, names of enzymes and names of bonds is almost my mantra. Yeah, learn them. The enzyme that forms the second strand of DNA, well, that's DNA polymerase. And the bond it forms, yeah, it's a good old phosphodiester. And crossing over is one way in which gametes can be made that are objectively different. Name one other way. Yeah, so that's independent segregation. Independent segregation, also called random assortment. You could actually put mutation there, but we're going to make that later on. Let's do the middle section. Again, you do require practicals, and therefore you should be picking things up during it, and you often do questions associated, and these are like kind of like taken from that a bit, but it's been a while since you've done it. So yeah, you have to know how to make a temporary mount from an onion root tip. We use garlic, same thing. And on the glass slide, she covered the tissue with a cover slip. She was then given the following instructions: push down hard on the cover slip, do not push sideways. Explain why she was given this instruction. So I need to know, she was then, yeah, push down hard, 1 mark, and not go sideways. Yeah, so she's trying to achieve a single layer of cells, not a thin layer of cells. Yeah, it's a single layer of cells. Yeah, to view chromosomes, or to view stages of mitosis. Remember, even if I have 2 cell thick, that's thin. But if one cell's over another cell, it's going to obscure itself from the other one. So yeah, a single layer of cells. And why not go sideways? Well, that would just like roll cells over each other or potentially damage chromosomes. Yeah. So it's easy to put like, may damage chromosomes. Again, I'm trying to view chromosomes. Last thing I want to do is damage them. That's that. Describe one other step that allows chromosomes to be seen with an optical microscope. Yeah, we have to stain them, not dye them. Yeah, we need to stain the DNA or the chromosomes. Yeah, we use a chemical stain, a histological stain that binds to DNA, and therefore it's visible. B part one's impossible for me to do because A, I don't have a ruler, and B, I'm on a screen that can change size, as I'm annoying you with now. So it's hard to kind of measure this scale bar. But let's assume that I've measured it, and it's, the image of the scale bar. I always measure the scale bar is, let's make it 10 millimeters. Always measure in millimeters. The actual size is 2 micrometers. The magnification is what I'm trying to find out, and therefore these units don't match. I need them in the same unit. Therefore, I'm going to convert the image, because it's easier to just multiply up than going to fractions of numbers. So yeah, the magnification equals, well, I need to get rid of the micrometer, so I need to divide these two numbers into each other, and magnification is going to be large, so it must be the big number on top, because otherwise I'd end up with a fraction again. And 2, yeah, again, micrometers and micrometers cancel, so it equals 500 times magnified. If I'm assuming it's a centimeter long, is the scale bar, and then it's like quite nice to be on the screen, because why is it preferable to have a scale bar when quoting magnification when publishing these images? Well, I can change the size of this. I've now got a more magnified image. My scale bar is now, let's say, 10 centimeters long. Therefore, I'm now magnifying it by 5,000. So you can rescale the image. So I write something along the lines that the image can be rescaled, and accurate magnification still made. Magnifications still calculated. The alternative is to just write on here times 500. But yeah, if I then zoom in, as I do, oops, just change my mode. Now it's obviously now not times 500. I've zoomed in and that still says times 500, whereas my scale bar is enlarged with the image. Again, that's a how science works, like common sense type thing. The stage shown is anaphase. It's a really obvious one, is anaphase, probably the most obvious. Then we get onto a mitotic index. There's an error here. This should say anaphase. I shall change it on your sheet, but I've not got it here. And yeah, the mitotic index, well, that's the number of cells in mitosis divided by the total number of cells. So if I've got 168 in interphase, so 200 minus 168 gives me mitosis. So it's, yeah, the mitotic index equals 32 divided by 200 multiplied by 100. Again, the number of people who divide by interphase is quite high. So yeah, 16%. And the mean length of the cell cycle is 36 hours. Calculate the time spent in metaphase. Well, I've got 5 out of 200 cells. So the number of cells is proportional to the time multiplied by 36. So yeah, I've got 0.9 hours. I can leave it like that as well. I don't have to change it into minutes and seconds and things. Just leave it like that. It doesn't say give the answer in minutes. So that's like required practicals. Yeah, how to make a temporary mount. And then there's a bit on meiosis. Yes, the diagram shows a pair of chromosomes at the start of meiosis. The letters represent alleles. What is an allele? Yeah, it's a form of a gene. There we go. Most genes are monoallelic, as in there's no variation within the species. Everyone's got the same DNA polymerase gene. There are no alleles of it, or there's only one allele of it. Whereas things like like for the gene that codes for our blood group antigens, then there are different alleles of that one. Explain the appearance of one of the chromosomes. Well, this is a chromosome has been replicated. Yeah, so it's a chromosome that's replicated. It's made of two things. So I'm going to describe this one here, but it could be the other one. And it's now made of sister chromatids joined by their centromeres. So each of these has a centromere. It looks like there's only one blob here. There's really two blobs squashed together. Okay. Just while I've got it, these are homologous chromosomes, but these are non-sister chromatids. Non-sister chromatids of a homologous pair. Just do a little bit of extra. Looks like a bit of free extra. If crossing over occurs at the locus of gene F, give the possible genotypes for genes E and F in the gametes made. So we're going to cross over here, move a big F onto the right-hand one and a little f onto the other one. Well, in terms of the gametes made, this chromatid here is going to separate away. So I'm going to get a big E, big F gamete. And then, oops, just change my pen. Get there eventually. Yeah, and then this one here, however, it's crossed over on the F, so I'm going to have a big E, little f gamete. Then I'm onto this one over here that's gained a big F, so it's little e, big F. And then I've got this one at the end over here, which is going to be, which has not done any crossing over, which is little e, little f. All we've done there is expanded the genotype, because this is if this is one organism, it's got a big E and a little e. It's heterozygous for gene E. It's just got two big E's and two little E's because it's copied its chromosome, and it's got a big F and a little f. So that's its genotype. Big E, little e, big F, little f. And then if I come across there, I get my big E, big F gamete, or I get my big E, little f gamete. I get my little e, big F gamete, and I get my little e, little f gamete. So I'm getting the same ones from its genotype, or I can get them from the diagram. And if it didn't make this one and this one, as in the ones that are recombinants, so those are recombinants. If it didn't make them, that would imply that genes E and F were very close together on the chromosome. It would look something like, so there's my chromosome. I'm going to put, there's gene E, and gene F is right next to it, and therefore they never separate. They're always right next to each other, and therefore I don't make the big E, little f gametes or the little e, big F gametes for this organism. Yeah, so a bit more on crossing over there, which you'd need to know for the two genetic stuff, dihybrid crosses. And then lastly, on this little section, apart from crossing over, give two other ways in which gametes are made that are genetically different. Well, we've got our independent segregation. Again, independent segregation. So that's when there's just that's random positioning of each homologous pair. Each homologous pair either side of the equator. So in humans, we've got 23 pairs of chromosomes. So in terms of the possibilities I get, it's 2 to the power 23 gives me the different possibilities that I can get just due to independent segregation. So yeah, that's 8.4 million to 2 sig fig. 8.4 times 10 to the 6 possibilities. And then if you add crossing over, then you get into ludicrously high numbers and are pushing it infinity. And you can also talk about mutation as well. And then just to finish off with some misconceptions, I'm going to keep it to my 15 minutes. Probably not, but close. I'm getting better at this. The first stage of mitosis is interphase. It's the first stage of the cell cycle. Yeah, mitosis equals PMAT. So be careful there. Cytokinesis is part of mitosis. No, it's not. It follows telophase. It's not part of telophase, but it might start happening within telophase, But cytokinesis is the splitting of the cell, that the dividing of the cytoplasm. So yeah, like when it starts and ends is a bit grey. Gametes undergo meiosis. Yeah, a really common one. That like sperm cells undergo meiosis. No, gametes, yeah, are made in meiosis. Made by meiosis. Yeah, this is from stem cells in e.g. the testes. So yeah, be careful there. Yeah, a gamete is a haploid cell. It's got half the original number of chromosomes. It can't undergo meiosis because it would never be able to like pair up either side of the equator because it's only got one set of chromosomes. So yeah, gametes are the product of it. And all cells complete the cell cycle during their life. No, they don't. Nerve cells don't. Yeah, so nerve cells stay in G naught. That's like an off branch of G1. So yeah, they just stay in the G1 before the S phase. They grow to their full size in the G1 phase, and then they just get parked in G naught because you don't want to replicate brain cells because that will disturb the connections between the existing brain cells, and that's what memories and functions are. So hopefully there was something there. Again, it might just be like a nod along, or yes, I know all this, but even that is like 20 minutes well spent. And it's not 20 minutes, it's 16, 17 minutes. So I'm going to have a celebratory coffee. Well done, mate.